Okay, let's unpack this. Anti-PD-1 immunotherapy. It's been, well, revolutionary for cancer treatment. I mean, a huge leap forward.
Absolutely. One of the biggest we've seen in decades.
But here's the kicker, the tough part. The majority of patients, and we're talking maybe, what, 70%? They still don't respond.
That's the challenge, yeah. A huge number still don't get the benefit we hope for.
And that huge resistance rate, it's really pushing researchers to look for the hidden reasons. Why isn't the immune system kicking in like it should?
Right. And historically, the focus has been very much on the immune cells themselves, T cells, macrophages, that sort of thing.
But today, we're digging into something different. Our sources point to a completely overlooked player, something non-immune. It's the nerves inside the tumor itself.
Exactly. We're diving into some really fascinating new findings that directly link something called cancer-induced nerve injury. Let's call it CNI, directly to why some patients resist anti-PD-1 therapy. Yeah, and this mechanism, it really suggests the nervous system isn't just, sitting there passively while the cancer grows. It's actively involved, potentially deciding whether the treatment works or not.
So active player, not bystander.
Yeah.
Before we get deep into CINI, though, we need to maybe set the stage with a related idea, paraneural invasion, PNI.
Good starting point, yes.
PNI. Clinicians, oncologists, they know this term, right? But usually it's just seen as a bad sign, a marker of aggressive disease. Can you explain what PNI actually is?
Sure. So PNI, paraneural invasion, is essentially what you see under the microscope. It's the physical evidence that tumor cells have infiltrated, moved into the nerves that are associated with the tumor. We call those TANs, tumor-associated nerves.
So cancer cells literally growing into the nerve bundles.
Precisely. And it's already well established as a hallmark of poor prognosis. You see it commonly linked with worse outcomes in aggressive cancers like cutaneous squamous cell carcinoma, cSCC melanoma, gastric cancer, pancreatic cancer, PDAC. If PNI is there clinically, it often signals trouble ahead.
Okay, so PNI is the visual clue. But our mission today, as you said, is to go beyond just seeing that invasion. We want to understand CNI, the functional damage, right?
Exactly, the actual injury.
How that injury messes with the immune system, and crucially, is there a way to maybe reverse it?
That's the goal. Understand the mechanism step by step and see if we can intervene.
All right, let's start with the clinic. The evidence from actual patients. Researchers looked at tumor samples. You mentioned cSCC patients. These were people in anti-PD-1 trials.
Yes, specifically a trial using neoadjuvant semiplimab, an anti-PD-1 drug given before surgery. They compared patients who responded well to the treatment versus those who didn't, the non-responders.
And what did they find? Looking just at that visual PNI sign at the start, before treatment.
The difference was stark, really predictive. The non-responders, the ones where the immunotherapy unfortunately failed, had a significantly higher rate of PNI right at baseline.
How much higher?
About 50% incidence in non-responders compared to just 15% in the responders, so more than 3 times higher. It established this really strong correlation. If the tumor was already tangling with the nerves, resistance was much more likely.
Okay, but hold on. PNI, as you said, often means the cancer is just more aggressive overall. How did they tease apart whether it was just general aggressiveness versus something specific about the nerve injury itself causing the resistance?
That is the critical question, and it's where the research really pivots. They moved from just looking at PNI, the invasion, to confirming CNI, the actual functional damage to the nerve cells.
How do you measure nerve damage? It's not like taking its temperature?
Right. They looked for specific biological markers inside the neurons themselves. Think of them as like molecular stress signals. And the most important one they focused on was a protein called ATF3.
ATF3. Okay, what does that tell us?
ATF3, activating transcription factor 3. You can think of it as the neuron's internal alarm bell. It's a gene, and its activity gets rapidly ramped up, upregulated, specifically when a neuron suffers physical injury or stress.
So it's the nerve cell essentially shouting, ouch, I'm hurt, trying to repair.
Exactly that. It signals damage and an attempt at neuroregeneration.
And connecting this back to the patients, were the nerves in the non-responders shouting louder, more ATF3?
Precisely. They isolated the neurons specifically, the beta-3 tubulin-positive, GFAP-negative cells from the tumor samples. And the neurons from the non-responding tumors showed significantly higher levels of ATF3 compared to neurons from the responders. The p-value is 0.005, so quite significant.
Wow. Okay. So the tumors that resisted the immunotherapy were the ones actively causing more nerve injury, triggering this ATF3 alarm.
Yes. It directly links the functional nerve injury state to the treatment failure.
This feels like it could be a bigger deal than just one type of skin cancer. Did they check if this holds true in other cancers? Make sure it wasn't just a cSCC quirk.
They did. That was crucial for establishing this as a potentially general or tumor-agnostic mechanism. They developed a gene signature based on PNI and these nerve injury markers like ATF3.
A kind of molecular fingerprint for this nerve injury process.
Exactly. And they applied this signature to large, publicly available data sets of patients with metastatic melanoma and gastric cancer who had also received anti-PD-1 therapy.
Did the signature predict resistance there too?
It did. In both melanoma and gastric cancer cohorts, resistance to anti-PD-1 therapy strongly correlated with this nerve injury signature. It really bolsters the idea that we're looking at a fundamental cross-cancer mechanism. CINI assay seems to drive resistance regardless of where the tumor started.
Okay, so let's recap this first part. We see a strong clinical link. Patients whose tumors show signs of invading nerves, PNI, are much less likely to respond to anti-PD-1.
Right.
And it's not just the invasion, it's actual functional nerve injury, marked by high levels of this distress signal, ATF3, inside the neurons.
Correct. The injury itself is key.
And this isn't confined to one cancer type. It looks like a general mechanism across melanoma and gastric cancer, too.
Seems to be, yes.
So the big question now is how? How does the cancer actually do this? What's the physical process causing this nerve injury?
Right. Let's zoom in on the mechanism. What's happening at that interface between the cancer cell and the nerve fiber?
They modeled this in the lab, I assume.
They did. They used co-culture experiments, essentially growing sensory neurons specifically from dorsal root ganglia, or DRGs, together with SCC cancer cells in a dish.
And what happened? Did the cancer cells interact with the nerves?
Very much so. The cancer cells were clearly neurotropic.
Meaning they're attracted to nerves.
Yes, they actively seek them out. Within just 72 hours, the cancer cells migrated towards the neurons and made direct physical contact with the axon, the main nerve fiber. It wasn't passive. It was like an active seeking and engaging.
Okay, they make contact. Then what? This is where it gets really interesting, right? The actual damage.
This is the core of CNI. Using very powerful electron microscopy, which gives incredibly high-resolution images, they pinpointed the primary mechanism. It's myelin degradation.
Myelin, that's the insulating sheath around the nerve fiber, correct? Like the rubber coating on electrical wire.
Exactly. It's a fatty layer that protects the axon and, crucially, allows electrical signals, nerve impulses, to travel quickly and efficiently. And the cancer cells were physically destroying this myelin sheath.
Not just damaging it, but destroying it. What did that look like?
Under the microscope, it was dramatic. They saw the myelin layers, which are normally tightly wrapped and compact, becoming disentangled. They saw myelin debris accumulating. In nerves exposed to cancer cells in the lab, you could see this process happening. After just 7 days, often there was a complete loss of the myelin sheath right at the point where the cancer cell was contacting the nerve.
Wow. So the cancer cell literally strips the insulation off the nerve wire. It's not just inflammation around the nerve, it's structural demolition.
That's a good way to put it, structural dismantling.
And presumably, stripping the insulation wrecks the nerve's function, its ability to conduct signals.
Absolutely. Structure dictates function here. They tested this using high-throughput electrical conduction studies in mouse models with these tumors.
Measuring how fast the signals travel.
Essentially, yes. They measured nerve conduction in the tumor-bearing skin compared to normal skin from the same animal. And the conduction, the signal strength, and potentially speed was significantly lower in the tumor area. It dropped from about 60 microvolts in normal skin down to about 42 microvolts in the tumor skin.
Nearly a third lower.
Yeah, a substantial drop. And that functional deficit, that slower, weaker signal, perfectly matches the structural damage, the demyelination they saw under the microscope.
Okay, this next point seems really important for figuring out potential treatments.
Yeah.
This damage, this myelin stripping, it happens because the cancer cell is physically touching the nerve, right? It's not just releasing some toxic soup.
Exactly. That's a critical distinction. They tested this specifically. They took the liquid medium that the cancer cells had been growing in, so it contained all the factors the cancer cells might secrete, but not the cells themselves.
The conditioned medium.
Right. And they exposed neurons to this conditioned medium. And the neurons remained healthy, no significant myelin damage. It really confirmed that the CINI, the injury, results from the direct cancer cell neuron contact and the physical process of myelin degradation. It's not primarily a paracrine effect driven by secreted factors from afar.
Okay, so direct contact, myelin stripping, electrical failure. This injury then forces the neuron to flip that internal ATF3 alarm switch we talked about.
Perfect.
But an internal alarm doesn't explain the resistance. The nerve must somehow broadcast this "I'm injured" signal to the surrounding area, to the immune cells. How does it do that?
It does exactly that. Once that internal ATF3 pathway is activated, the injured neuron itself starts acting like an immune signaling cell. It autonomously releases a specific set of immune-related molecules, inflammatory signals.
The nerve itself becomes an immune regulator.
In a sense, yes. It's responding to the injury by sending out signals that recruit and modulate immune cells.
What kind of signal? What are the key messages it's broadcasting?
They measured various cytokines and signaling pathways. The ones that stood out, showing the most prominent increases after the neurons were exposed to cancer cells, were pathways related to interleukin-6, IL-6, and type I interferon, or IFN-I signaling. Things like TNF-alpha and IFN-gamma were also involved, but IL-6 and IFN-I seemed particularly key.
And crucially, where were these signals coming from? The injured nerve or the cancer cell?
This was important. They confirmed that IL-6, for instance, was specifically secreted in much higher amounts by the neurons after they'd been exposed to the cancer cells. It wasn't the cancer cells suddenly pumping out more IL-6, it was the injured nerve itself generating this inflammatory signal.
So the injured nerve is basically screaming help, using IL-6 and IFN-I signals, trying to initiate some kind of repair process in the tumor microenvironment.
That's the likely intent of the pathway, yes, an injury response.
Which leads us to the really puzzling part. If it's an inflammatory healing type signal, why does it lead to resistance against the anti-PD-1 therapy? Why doesn't it help fight the tumor?
That's because this isn't a normal transient injury. When you have a peripheral nerve injury, say, from a cut, it does attract immune cells to promote healing and repair. That happens in what we call the perineural niche, the area right around the nerve.
Okay, a normal healing response.
But here, the injury isn't transient. It's being caused by a continuously growing, invading tumor. So the CNI is chronic. It's sustained. And that chronic injury signal gets completely corrupted, hijacked by the tumor environment. Instead of promoting a helpful acute inflammation that clears debris and aids repair, this chronic signaling driven by the injured nerve actually fosters an immunosuppressive environment. It leads to immune exhaustion and suppression rather than an effective anti-tumor fight.
So the nerve's chronic cry for help inadvertently tells the immune system to stand down.
In effect, yes. CINI transforms the tumor microenvironment, especially that local perineural niche, into an immunosuppressive landscape. The researchers found that those injured nerves, the ones positive for both nerve markers like NFH and the injury marker ATF3, were tightly correlated with an increase in specific types of suppressive immune cells.
Which types?
Particularly immunosuppressive macrophages, often called M2-like macrophages. These ones expressed CD68, CD163, and PD-L1, and also functionally exhausted CD8 plus T cells, the main cancer-killing cells, marked by TIM3 expression.
And this suppression is geographically focused, at least initially, right around the nerve.
It starts there, yes. Very concentrated in that perineural niche, the immediate vicinity, say, within 150 micrometers of the injured nerve fiber. They used spatial transcriptomics, a technique that maps gene activity in tissues.
Cool tech. What did it show?
It confirmed that areas right around injured nerves, those expressing both ATF3 and another injury marker, JUN had significantly higher infiltration of those CD163 plus suppressive macrophages, and also LEG3 plus exhausted CD8 plus T cells, compared to areas around healthy, uninjured nerves in the same tumor.
So a little bubble of immunosuppression forms right around the damaged nerve.
Exactly, a localized suppressive niche.
And that must be a major reason why the anti-PD-1 therapy fails, right? You can't overcome that local suppression.
That's certainly part of it, but the problem doesn't stay localized. As the tumor continues to grow and more nerves get invaded and injured, they observe this over time in mouse models, the CINI burden increases until it sort of saturates around day 11, that nerve-driven inflammation becomes a chronic, pervasive process.
It spreads beyond the bubble.
Yes. The persistent immunosuppressive activity that starts in the perineural niche eventually begins to skew the overall immune tone of the entire tumor microenvironment. It dampens anti-tumor immunity more globally within the tumor. They found correlations in the levels of suppressive cells like CD8 plus LAG3 plus and CD163 plus PD-L1 plus between the perineural niche and areas far away from any nerves within the PNI positive tumors.
So the local problem poisons the whole guess. Well, immunologically speaking. The T cells might be there, drawn to the tumor, ready to fight.
They often are present, yes.
But that chronic alarm signal originating from the injured nerves has basically drained their batteries, made them exhausted and unresponsive.
That seems to be the critical outcome. It leads to immune exhaustion. When they looked back at the clinical samples, the non-responders didn't necessarily lack T cells in their tumors. But the T cells they had were functionally impaired. They showed significantly higher overall expression of PD-1, the very target of the therapy, suggesting they were more exhausted.
So the therapy fails not because the target isn't there, but because the T cells are too worn out, largely thanks to this chronic nerve injury signal.
That's the conclusion. The nerve-driven chronic inflammation appears to be a core reason for that T-cell exhaustion and the resulting resistance to anti-PD-1.
Okay, wow. That paints a really clear step-by-step picture from nerve invasion to treatment failure. It's quite a stunning display of causality.
It really connects the dots in a new way.
Which brings us to the most hopeful part, maybe. If we've established that CNI causes resistance, can we stop it? And if we stop the CNI or its consequences, does the anti-PD-1 therapy start working again? Can we reverse the resistance?
Yes. And this is where the evidence gets really compelling. The researchers tested this directly and found that, yes, this process seems to be reversible. They demonstrated this in several ways, providing multiple lines of evidence by targeting different points in that cascade we just described.
Okay, let's hear the proofs. What was proof number 1?
Proof 1 was perhaps the most direct approach. Just get rid of the nerves altogether. They performed a denervation procedure in their mouse tumor models.
Surgically cutting the nerves, supplying the tumor area.
Exactly. Removing the nerves that would potentially become injured. And the result. In these denervated mice, the anti-PD-1 therapy worked significantly better. Tumor control was enhanced compared to control mice that had a sham surgery but kept their nerves intact. The p-value was 0.00027, a strong effect.
So no nerve, no CNI, no resistance signal. Immunotherapy works better. Makes sense.
It's strong causal evidence.
Okay, proof 2. This one sounds more subtle, targeting the alarm inside the nerve cell, that ATF3 signal.
Right. Instead of removing the whole nerve, they used a more sophisticated genetic approach. They created a conditional knockout mouse model, let's call it ATF3-CKO, where they could specifically delete the ATF3 gene only in the relevant sensory neurons, the nociceptors, the ones that sense pain and injury.
So the nerve is still there. It might still get damaged by the cancer, but it can't flip that internal ATF3 alarm switch.
Precisely. They impaired the core injury signaling within the neuron, and the results were striking. Just knocking out ATF3 in those neurons led to reduced melanoma tumor volume on its own.
Even without immunotherapy.
Even without it. And when combined with anti-PD-1, it significantly improved the anti-tumor activity. They saw more activated CD8 plus T cells producing interferon gamma, which is a key anti-cancer cytokine.
And what about those suppressive macrophages? Did blocking ATF3 affect them?
It did. In the mice lacking the ATF3 signal in their neurons, the macrophages within the tumor shifted their phenotype. They became less like the immunosuppressive M2 type found in the normal injured setting and more like the anti-tumor M1 type. It basically reprogrammed the immune environment just by silencing that one nerve signal.
Incredible. So targeting the nerve's internal response directly counters the suppression that really pins ATF3 as a central mediator.
Absolutely. It shows ATF3 isn't just a marker, it's functionally driving the immunosuppression.
Okay, 2 proofs down. Denervation and blocking the internal alarm. What was proof number 3? This targeted the signals the nerve sends out, right? The IL-6 and IFN-I.
Exactly. They went after the downstream consequences, those inflammatory cytokines being released by the injured neuron. Could blocking them restore sensitivity to anti-PD-1?
Let's start with type I interferon. How did they block that?
They used mice that were genetically engineered to lack the receptor for type I interferon IFN-I mice. So even if the injured nerve released IFN-I, the surrounding immune cells couldn't receive the signal.
And did that prevent resistance?
It did. They induced nerve injury in these mice using a chemical agent that causes demyelination, mimicking CNI. In normal mice, this demyelination caused resistance to anti-PD-1 as expected. But in the IFN-I mice, the anti-PD-1 worked just as well, whether the nerves were demyelinated or not. Blocking IFN-I signaling completely negated the resistance caused by the nerve injury.
Which supports the idea that the chronic IFN-I signal from the nerve is actually immunosuppressive in this context, right? Not pro-immune.
Correct. In this chronic setting, it appears to drive suppression.
Okay, and what about IL-6? You mentioned that was very prominent. Did blocking IL-6 also work?
Yes. They combined anti-PD-1 therapy with an antibody that blocks the IL-6 receptor, anti-IL-6R. This combination therapy significantly reduced tumor viability and restored the effectiveness of anti-PD-1, but only in the mouse models where they had induced demyelination mimicking CINI.
Ah, so the benefit was specific to the nerve-injured tumors.
Exactly. If the tumor didn't have that underlying nerve damage, blocking IL-6 didn't add much benefit to the anti-PD-1. This strongly suggests the IL-6 they were successfully targeting was indeed coming from the CNI-driven inflammation originating from the injured nerves, not just some general IL-6 from the tumor cells themselves.
So 3 different approaches, all converging on the same conclusion. You can reverse this CII-driven resistance.
That's right. You can target the nerve itself by removing it. You can target the neuron's internal injury response by blocking ATF3, or you can target the downstream inflammatory signals like IL-6 or IFN-I signaling. All 3 strategies appear to restore sensitivity to anti-PD-1 therapy in these models.
Let's try to wrap this up then. This deep dive, it's really uncovered something quite profound. The nervous system isn't just passively affected by cancer, it's actively regulating the immune response to it.
It's acting as a direct immunoregulatory agent.
And this specific mechanism, CINI, where cancer cells physically degrade the nerve's myelin sheath, forces the neuron to send out a chronic alarm signal.
Driven by ATF3 upregulation, leading to that sustained release of IL-6 and type I interferon.
Which, instead of helping, actually attracts suppressive immune cells, exhausts the killer T cells, and ultimately erects this barrier, making anti-PD-1 immunotherapy ineffective for many patients.
That's the cascade, yes. From physical nerve injury to functional immune suppression and treatment resistance.
So what does this all mean? For you, listening, for researchers, for potential future treatments, it feels like a big shift in perspective.
I think it is. These findings really open the door to completely new therapeutic strategies. We spent so long focusing on targeting the tumor cells directly or targeting the immune cells directly. Now, maybe we need to target the interaction between the nervous system and the immune system within the tumor.
Treating the nerve to treat the cancer, essentially.
Potentially, yes. Or at least modulating that nerve response. It also gives us potential new biomarkers.
Right. You mentioned checking for P&I at baseline or maybe measuring ATF3 levels in biopsies. Could that help predict upfront who is likely to resist standard anti-PD-1?
That's a definite possibility. Identifying these patients early could allow us to try different strategies, perhaps combining anti-PD-1 with one of these nerve-targeting approaches.
Blocking IL-6, for example, seems clinically feasible since anti-IL-6 drugs already exist.
Exactly. It provides actionable targets based on this new understanding of the biology. Instead of just trying to push the accelerator on the immune system harder and harder, maybe the first step for some patients needs to be releasing the brake that the injured nervous system is applying.
Calming the nerve signal first. Which leads to a final thought, maybe a provocative question for everyone listening. If this chronic inflammation, driven by nerves under attack by the tumor, is such a fundamental block to effective immunotherapy, should the next big leap in cancer treatment actually come from neuroimmunology?
It certainly puts neuroimmune interactions squarely on the map for oncology.
Specifically, maybe silencing that pain or injury signal that the tumor forces the nerves to broadcast. When we think about attacking cancer, we think about killing the tumor cells or boosting the immune system. But maybe we need a third pillar, calming the hijacked nervous system within the tumor. Which approach should come first, or should they be simultaneous?
That's the complex question going forward. Are these processes, tumor growth, immune response, nerve signaling, so intertwined that we absolutely have to address all 3? Today's research certainly suggests that ignoring the nerve component might mean leaving a major driver of resistance untouched.
Something to definitely keep thinking about. Fascinating stuff.